Hello everyone and welcome back to Easy Dentistry, a channel that makes dental education incredibly easy and interesting. If you have not yet done, please subscribe to our channel right now. Also, if you like our lectures, please let us know your feedback in the comment. And most important, please don't forget to share these lectures with all your friends and share knowledge. We have started uploading lectures on the subject microbiology. You can find the lectures on microbiology in a separate playlist on our channel page. In the previous lecture, we had studied the bacterial growth, and most importantly, we have studied the bacterial growth curve. Hope you have watched that lecture. In continuation with the previous lecture, this lecture is on bacterial nutrition, in which we will be studying the factors which influence the growth of microorganism. The factors. The factors influencing the growth of microorganism is an important 10 marks question which is asked in your exams. So watch this lecture carefully. Now, the principal constituent of a bacterial cell is water. The water constitutes around 80% of the total weight. For growth, bacteria need most importantly water, then carbon, nitrogen, and inorganic salts. In addition to these requirements, it also requires certain elements like sulfur, potassium, magnesium, iron, and manganese for bacterial growth. And some bacteria are very selective in their growth and will not grow unless certain organic compounds are added to the medium. These organic compounds are called as growth factors or bacterial vitamins. The bacterial vitamins are of two types, the essential vitamins and the accessory vitamins. The essential growth factors or the essential vitamins are those which are necessarily or essentially required for the growth of bacteria. If these essential vitamins are not present, bacteria will not grow. But accessory vitamins are those vitamins which are not essential but they enhance the growth of the bacteria. Other vitamins which are needed are thiamine, nicotinic acid, riboflavin, pyridoxine, folic acid, and vitamin B12. These are the vitamins of the vitamin B group complex. Now, the clinical significance to know these essential factors for bacterial growth is for the preparation of culture media. For the growth of specific bacteria, you need to add specific nutrients to the culture media. Now, the environmental factors affecting growth. The important long answer question. And in this part of the lecture, you will also find numerous small terminology which are asked in your MCQs. So, the first factor which affect the bacterial growth is moisture and desiccation. So, moisture is an essential requirement for the growth, because 80% of the bacteria is nothing but it is water. So moisture or water is essential. However, drying or desiccation has varying effect in different organisms. Now, Treponema and Neisseria, they die quickly on drying, while Mycobacterium tuberculosis and Staphylococcus, they survive for several weeks. So there is varying effect of drying. Now, clinical significance of knowing this moisture and desiccation effect on bacteria is as Neisseria and Treponema are susceptible to drying and they die or if desiccated, so they require a special transport media to transport the clinical specimen. On the other hand, as Staphylococcus are not susceptible to desiccation, so Staphylococcus does not require any transport media. Also, lyophilization is an important method for preservation of microorganisms. Lyophilization is nothing but freeze drying, and in which drying cold and vacuum is used for preservation. The second factor affecting the growth is the oxygen level. On the basis of oxygen requirement, there are two types of bacteria, the aerobes and the anaerobes. The aerobes, they require oxygen for their growth. There are two types of anaerobes, the obligate anaerobes and the facultative anaerobes. The obligate anaerobes, as the name suggests, the presence of oxygen is obligatory for their growth. That is, they grow only and only in the presence of oxygen. Example of obligate anaerobes, obligate aerobes are Pseudomonas aeruginosa. The it requires oxygen and for its growth. Oxygen presence is obligatory for the growth of Pseudomonas aeruginosa. Then comes the facultative anaerobe. These facultative anaerobe can grow without oxygen. Most of the pathogenic bacteria are facultative anaerobes. Then comes a terminology called as microaerophiles. The microaerophilic bacteria can grow in the presence of traces of oxygen. Even if oxygen is present in traces, they can grow. Example of microaerophiles are Helicobacter pylori and Campylobacter. Then what are anaerobes? Anaerobes are the bacteria which grow in the absence of oxygen. The example of anaerobes is Clostridium tetani. Even if oxygen is present in traces, the anaerobes do not grow. Then the third factor is carbon dioxide. The small amount of carbon dioxide is required for the growth of all bacteria. The carbon dioxide may be present in atmosphere, or there may be endogenous production of carbon dioxide by the bacterial cell. Now, what is capnophilic bacteria? The capnophilic bacteria require more level of carbon dioxide than other bacteria for their growth. They require around 5 to 10% of carbon dioxide. Example of capnophilic bacteria is Brucella abortus. So, remember here the capnophilic bacteria and its example. The fourth factor influencing growth of the bacteria is the temperature. Now, what is optimum temperature? Optimum temperature is the temperature at which the growth of the bacteria occurs best. It varies from species to species. Most of the pathogenic bacteria grow well at 37 degrees Celsius, that is at body temperature. Depending upon the varying optimum temperatures, there are three groups of bacteria: mesophiles, psychrophiles and thermophiles. The mesophiles, they grow well between 25 to 40 degrees Celsius temperature, and majority of the pathogenic bacteria are mesophiles. Psychrophiles, they grow well below 20 degrees Celsius, and examples of psychrophiles are soil and water saprophytes. Then comes the thermophiles, which grow between 55 to 80 degrees Celsius. Example of thermophiles is Geobacillus styrothermophilus. So remember the example of thermophiles. This is asked frequently in your MCQs. Geobacillus styrothermophilus is a thermophile. This bacillus was formerly known as Bacillus styrothermophilus, but the new name is Geobacillus styrothermophilus. Now, the clinical significance of temperature is that most of the pathogenic bacteria, they die between 55 to 65 degrees Celsius, and the spores die between 100 to 120 degrees Celsius. So, the knowledge of the temperature at which the bacteria die is useful for the sterilization. Also, low temperatures are useful in preservation of the bacteria. The bacterial cultures are stored in refrigerator at 3 to 5 degrees Celsius and in deep freeze in -30 to -70 degrees Celsius. Then the fifth factor comes the pH. Most of the medically important pathogenic bacteria grow well at a pH of 7.2 to 7.6. Some of the bacteria such as lactobacilli, they grow well in acidic pH, while Cholera Vibrio, they grow well at alkaline pH. If exposed to strong acid or strong alkali, the bacteria are killed, but the mycobacterium are resistant to these strong acids and alkalis as well. Now, the clinical significance is that the culture media having alkaline pH are used for growing Cholera Vibrio. Strong solution of acids or alkali are used in Petrov's culture medium, Petrov's concentration method for sputum specimen for it contains strong acid and alkali, which is favorable for the growth of Mycobacterium tuberculosis. Sixth factor is the light. The bacteria grow well in darkness and they are sensitive to UV rays and other radiations. Phototropic species are those which grow more in light, while photochromogenic Mycobacterium, they produce pigment when exposed to light. Then comes the seventh factor, which is osmotic effect. Bacteria are able to withstand a range of external osmotic variation because of the mechanical strength of the cell wall. What is plasmolysis? Plasmolysis is the sudden exposure of bacteria to hypertonic solution. This causes shrinkage of the bacteria and withdrawal of water from the bacterial cell. Plasmolysis, gram-negative organisms are more prone to plasmolysis than gram-positive ones. What is plasmoptosis? Is excessive imbibition or uptake of water by the bacterial cell. Plasmoptosis causes swelling and rupture. The plasmoptosis occurs due to sudden transfer of the bacterium from concentrated solution to distilled water. Then the last factor affecting the growth of bacteria are mechanical and sonic stresses. Vigorous shaking with glass beads causes rupture of the bacterial cell wall. Also, exposure to ultrasonic vibrations causes rupture of the bacterial cell. The next lecture will be on bacterial metabolism, in which we will be studying the oxidation process, fermentation process, and oxidative reduction potential. That's all for now. Thanks for watching.